Hello, my name is Eddie Guerra, and I will be proposing an additive manufacturing method of manufacturing polymer-based stents for safer vascular care. So, the specific problem I'm looking to tackle is ischemic heart disease, which is characterized by narrowed arteries that restrict blood flow to the heart. Now, this is a bit of an umbrella term, but one of the most common versions is coronary heart disease, which is pictured to the right. In the unhealthy artery, plaque builds up, causing impeded blood flow. Now, ischemic heart disease is a massive problem around the world. In the United States, it's estimated that $80 billion is spent combating the disease, and 11.6% of all Medicare patients have some form of it. One study showed that in 2019, 16% of all deaths around the world were connected to this heart disease. Now, there are many current solutions, some of which I have listed here. There are medications like antiplatelets, which prevent clotting, and pressure medicines, which reduce workload on the heart. Stents are meshes that open up a blood vessel to reverse that narrowing. One can also make lifestyle changes such as quitting smoking, improving diet, or getting better exercise. A more invasive solution here is the artery bypass, where a blood vessel graft is taken from another part of the body and blood flow is routed around the blockage using that vessel. There are devices that can monitor conditions and devices like defibrillators that can be used in extreme circumstances. And then finally, there are new emerging technologies like gene therapies and stem cell therapies that are being researched to alleviate the burden of this disease. Now, I want to focus specifically on one of these solutions. I want to focus on the stent. As I mentioned, these are meshes, and they're most commonly and historically made of metal. They're inserted with a balloon, which is blown up to expand the stent, and then it sits in place to open up the narrowed artery. This allows for prevention of collapse or further blockage of that artery, which in turn improves circulation, blood function, and reduces angina or chest pain. Now, there are a couple of types of stents, each with their own characteristics and drawbacks. The first is the bare metal. The first is the bare metal stent, which is the most primitive. This is just a metal mesh with no coating or anything on it. They're made of biocompatible metal alloys. So you can imagine with just a bare metal mesh sitting in a blood vessel over time, it can cause scarring. And it also has the highest rate of restenosis of all types of stents, which is a re-narrowing of the vessel after stenting. Over time, scar tissue can grow, and a bare metal stent is not equipped to handle that. So the next one is the drug-eluting stent, which has the bare metal stent as a base, but the metal is coated with a polymer that releases a drug which can stop the scar tissue growth found in restenosis. However, since the polymer is only a coating, there's a limited amount of drug and thus a limited lifetime for the stent. And then lastly, there are bioresorbable stents or biodegradable stents that break down over time while eluting drug, so they clear out of the system after the vessel is healed without the need for removal. The problem with these is that they are very new and very prone to mechanical issues. They have a hard time retaining enough strength to open or to hold open the vessel, especially when significantly degraded. The polymeric stent to the right there had very small defects, which caused its developmental failure due to mechanical failure. So the objective here is to develop a viable, i.e., mechanically viable polymeric stent, which can be loaded with anti-inflammatory or other relevant drugs via vat polymerization. Now, just as a side note, an important key idea here is that this needs to be presented to a company that would be willing to hear us out based on alignment of their business focus and the nature of our idea. So given that this would be an additively manufactured cardiovascular device, there are two types of partnerships that could work. One is a company focused on cardiovascular medical devices with maybe a bit of additive manufacturing experience. And then the second is a company focused on additive manufacturing for medical devices with maybe a bit of cardiovascular device experience. Just because a company that specifically focuses on additive manufacturing for cardiovascular medical devices, that's just a bit niche, at least as far as my research took me. But one or the other would be sufficient. Now, here to the right are some of the places that just fit into one of those criteria based off my search. And I ended up going with 3D Systems. They're a medical device company focused, or they're medical device focused additive manufacturing company. And I found that they have specific expertise in vat polymerization, which is super useful for this project because since I'm using vat polymerization, it would cut down on costs for equipment, maybe even training, among other things. So now moving into why I chose vat polymerization. So the schematic to the left here shows a specific type of vat polymerization known as stereolithography. Vat polymerization is also an umbrella term, but stereolithography is abbreviated to SLA. Um, and the process here is that a vat, as the name suggests, is filled with resin, and this resin contains a photo initiator, a photocurable monomer, and any additives needed, like plasticizers or stabilizers. Then a UV beam selectively polymerizes the first layer by shining on specific points on the layer according to the CAD design. That UV light is able to activate the photoinitiators and causes polymerization and curing of the polymer because the polymer is responsive to light. Once that layer is complete, the platform holding that layer moves down, allowing more resin to kind of collect on the top, forming a second layer, and then photopolymerization continues. Basically, the first layer that's formed stays on the platform, and the last layer ends up being the highest one in the vat. Once all the layers are complete, the finished product is removed, rinsed, cured, and any post-processing can take place. So again, why vat polymerization? There are a couple of advantages here when it comes to building a stent. First, it has a resolution of down to 20 microns, which is very high resolution compared to some other additive manufacturing techniques. It's really only limited by the beam width, the UV beam width. This allows for high control over the thickness of stent struts and just the overall geometry of the product. The drug can be added to the resin vat, so it can be homogenized in the resin, and there won't really be the shearing problems that might be seen in extrusion, for example. Because CAD is used to design the product, stents can be highly tailored to the needs of the patient, which is important because every patient is different in almost all cases. Also, this is quite innovative in terms of stent manufacturing because most additively manufactured stents, at least right now, um, are metal, and those that aren't are typically made through extrusion. And then lastly, there are several available photopolymers that are available for the processing here. But the photopolymer I chose is polyethylene glycol diacrylate, which I'll just call PEGDA for simplicity. Obviously, it has to be photocurable, so it polymerizes on contact with the UV light. It's also PEG-based, so it's biocompatible, and it's highly tunable, meaning it's flexible. Flexibility, cross-linking density, and other properties can be easily altered through different processing techniques. And then lastly, it's well known for its ability to work as a vehicle for drugs. It's able to contain them well and also release them in an appropriate manner. So here is my proposed solution process. First, the drug is dissolved in an organic solvent so that it can be incorporated into the resin. I chose rapamycin because it's commonly used in the drug-eluting stents, but it inhibits smooth tissue cell proliferation, so it reduces scarring and restenosis that I was talking about earlier. This is then mixed with the monomer, the photoinitiator, and any additives to make the resin. At this point, the resin can be conjugated with agents that improve its biodegradability. PEG itself isn't really known for biodegradability, so drug delivery would be largely based on diffusion. I will say that it has been found that PEG with the diacrylate does exhibit some safe biodegradation. However, with the addition of degradable bonds, such as hydrolyzable crosslinks, the stent could be made to be more biodegradable to increase release. Basically, if the stent can degrade better, the drug is released quicker. But this isn't really in the scope at this point. This could be kind of a secondary project to the main one. And then at this point, any excess organic solvent from the rapamycin mixture can be evaporated off, but organic solvent from the resin will also be evaporated during cooling or curing, rather. So next, the SLA process, as I described earlier, can take place, assuming there's already a CAD model for the stent that the equipment can follow. Once the printing is complete, simply clean the stent with something like IPA and do the final curing with more UV light just to solidify the stent. Now, this last step here is again optional. So other polymers such as PLGA, which is FDA approved, can be added to the surface to affect release. If this coating, let's say it's PLGA, also contains drug like a drug-eluting stent does, there could be a sort of burst release or quicker release overall. If not, it will mean if the drug isn't in that coating, it will mean there's more polymer for the rapamycin to diffuse through, slowing the release. Again, this could be treated as a second project as the primary scope here is developing the polyethylene glycol diacrylate polymer through SLA. So just with any other research, the stent will need to go undergo some verification testing. So first we need to ensure that the structure we want was actually synthesized. FTIR and NMR will confirm that rapamycin is successfully incorporated and that the polymer didn't undergo any molecular changes during synthesis. SEM can show surface morphology and any defects that might affect the stent's efficacy. It's also important to know what forces the stent can withstand in the blood vessel and for how long, since a stent would experience stresses over time. So, according to a study at Kyushu University in Japan, a stent needs to withstand 1.5 newtons of compressive radial force around it in order to not collapse. And that's for a single load, so it would be different for cyclical loading or fatigue testing. But that 1.5 Newton figure is viable for this polymer. So to test this again, we would do compression testing and cyclical loading tests. Now, swelling ratio is also important to know because it has implications on how well it's going to be able to hold open the blood vessel. If it's able to swell well, it'll be able to hold open that blood vessel. And it also just affects drug release, the way that a polymer swells. HPLC and UV spectroscopy can verify the pharmacokinetics for in vitro drug release testing. And then the hemocompatibility test will ensure that the stent doesn't cause blood clotting or other adverse effects. And then lastly, to study biocompatibility, the MTT assay studies cell viability when exposed to the stent, and the inflammatory response test will see if an inflammatory response is caused when the device is exposed to immune cells or macrophages. So now to go over the project timeline. Overall, we're aiming for a 6-year development process with milestones all along the way. Year one is obviously early stage and brings researching and prototyping milestones. Two of the things we'll be looking for here are actual fabrication of the stent, along with concept feasibility analysis, just to ensure that this is a project that's worth spending our time, money, and efforts on. This stage will be characterized by preclinical development and in vitro testing. Year two will bring several development milestones. We'll be looking to optimize the stent, especially investigating, including any additives to the resin and seeing if those hydrolyzable crosslinks or polymer coatings I mentioned earlier need to be added. We would be heavily focusing on the design verification tests. And we would continue with preclinical testing, especially animal trials and in vitro testing. So years 3 and 4 will hopefully bring big leaps here to the development of the device, as this period will bring the start of clinical investigations. First, we'll need to obtain the FDA investigational device exemption, which basically just grants us the ability to perform clinical trials. And then we can enroll patients and start a phase 1 clinical trial to assess the safety of the product. This will allow us to further validate the device, and this time period could also include the start of a phase 2 clinical trial if phase 1 goes well. And then lastly, years 5 and 6 can bring us to our fourth milestone of commercialization. The phase 2 clinical trial to test feasibility in actual IHD patients will have to be completed. And then the phase 3 clinical trial to test on a large population will have to be started and completed also in this time frame. Additionally, more regulatory paperwork will have to be filled out, and we'll have to work on scaling up the product. And then lastly, we'll need to obtain pre-market approval, launch, and market the product. Now, of course, it's entirely possible that things don't go according to plan. In fact, that's expected, really. And development has to if development has to last more than 6 years, that's okay. But this is certainly a doable endeavor here in 6 years. Now moving to the cost estimate. In total, we'll be looking for $10.5 million to fund this project. Over 6 years, this will cover personnel, development, and preclinical research, clinical trials, facilities, and manufacturing scale-up, and regulatory and market entry. Of course, this is contingent on the availability of certain equipment and materials at 3D Systems. Now, obviously, there are going to be some risks and drawbacks that we could stumble upon along the way. One such risk is the fact that polymer stents historically have issues with mechanical properties. While the polyethylene glycol diacrylate does have impressive mechanical properties compared to some other polymers that have been researched, there's a very real possibility that this will be a significant hurdle and changes in formulation will have to be made. This leads to the second point, which is the fact that, as mentioned before, there may need to be changes in the fundamental formulation and coatings if mechanical properties or drug release do not live up to standard. These changes will cost money in research and materials if they need to be made. Third, stents are small, so printing intricacy is a risk. The geometry has to be very precise there. And then lastly, stents can be difficult to insert, especially because every patient is different. So the customizability of these stents has to be confirmed. As for external risks, we are obviously at the mercy of the FDA. We have to follow guidelines, and changes they request are non-negotiable. This device also relies on the feedback of key opinion leaders and clinicians, and if they don't view this device as viable, it could mean the end of the project. Lastly, in a changing world, supply chain dynamics can cause issues in many different ways, from resin component availability to transportation costs to increasing lead times. Now, I've mentioned a couple of times that the potential that the resin may need to include additional components such as stiffer polymer blend or hydrolyzable crosslinks. This has been built into the budget if need be. And then lastly, for kill criteria, if mechanical irregularities continue even after those adjustments, if drug release is not consistent and controllable, or if the FDA rejects the idea, project will have to come to an end. So to summarize, this project includes carrying out vat polymerization of rapamycin-containing polyethylene glycol diacrylate stents. This is aimed towards the company 3D Systems and utilizes SLA to polymerize specific areas of a resin in a vat with UV light to make a print. The polymer is key in this project because is photosensitive and biocompatible, and it has tunable material properties. Finally, the drug used is rapamycin, which reduces inflammation and restenosis. So the advantages of this process include high resolution from the SLA process to create an intricate design, as well as a homogeneous drug in the resin. The CAD printing aspect means the stents would be customizable, and stents are currently not made in this fashion in the market. Since this is a polymer-only base as opposed to metal, it can load more drug than those metal-based stents. And then finally, it is able to achieve mechanical strength through polymer swelling. Here are my references, and thank you.